Hello, and welcome to this video lecture for Microbiology Bio 203, Chapter 9 of your OpenSTAX book on microbial growth. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. Learning outcomes for this chapter include being able to describe the phases of microbial growth and quantify that growth. We begin today by examining exactly how microbial growth is defined. Microbial growth is a reflection of an increase in number of cells in a population or culture, not in an increase in cell size. The most common way microbial cultures grow is through binary fission. Binary fission is very similar to, but not identical to, mitosis. In binary fission, the DNA replicates, then the rest of the cell divides, starting in the middle of the cell, finally resulting in two separate cells. That process is illustrated at the bottom of this slide. Next, we'll look at generation time. The generation time for a microbial culture is the amount of time it takes for one cell to become two cells, or two cells to become four, and so on. Simply put, it's the amount of time it takes for whatever population you currently have to double in size. The pattern of cell doubling is illustrated for you here on this slide. Binary fission produces growth in exponentials of 2. The shorter the generation time, the faster the population is growing. Your generation time depends on the individual microbe and the conditions under which it is growing. This table provides a graphic illustration of exactly how fast a single cell can become literally millions of cells. What's left out of this table is the exact time period of the generation. The bacterial growth curve illustrates the phases through which cells travel as their population grows and potentially subsequently declines. Here you can see a lag phase for a population as the cells within the population show no net increase in number. Then a log phase or exponential growth phase follows. After the exponential growth phase, as resources begin to become limited in the environment the cells are growing, the cells will reach a stationary phase. From an ecological standpoint, we often refer to this stationary phase as the carrying capacity for the environment that the cells are in. Following this carrying capacity or stationary phase, a death phase or decline phase will follow as resources become so limited and the environment polluted with the waste products of the cells within it, that death follows. The lag phase. Here is a little bit more information on the lag phase. The bacteria will take some time to adapt to their new environment before actively dividing. Bacteria may not be in a growth phase when they're introduced into a medium. They may be introduced as spores. The bacteria may be utilizing different biochemical pathways for growth in other medium. The bacteria may experience physical changes upon introduction to a new medium, all things that cause growth to be delayed. The log or exponential phase. Here, the bacteria divide regularly. The idea behind log or exponential growth is that their rate of growth increases and the time it takes for that growth decreases. The stationary phase, as mentioned before, is a phase where nutrients begin to become exhausted. Waste products begin to accumulate. pH may also be changing in the medium the organisms are growing in. All things considered, the environment is not as supportive as it once was, and the population reaches a point where the number of cells made is roughly equal to the number of cells that are dying. I described this a moment ago as carrying capacity from an ecological standpoint. Here we have the death phase. This is logarithmic decline. At this point, the environment will no longer support life. Some cells may survive through the production of endospores. At the end of the day, if nothing is done, the entire culture will die. Next, we get methods of quantification of bacteria. There are direct and indirect methods that can be used to quantify how many microorganisms we have in a population or culture. The direct methods involve plate counts, filtration, most probable number assessments, and direct microscopic counts, while the indirect methods quantify the number of organisms based on turbidity, metabolic activity, and dry weight. This is the direct microscopic count. Here we use a microscope and special slides with grids. The slides hold an exact volume between the cover slip and the slide surface. A researcher would literally count a bacterial dilution. This unfortunately does not distinguish between live and dead cells. It only counts how many cells are present. Plate counts can also be used in direct quantification studies. A pour plate involves using warm agar to dilute the bacteria. We then mix and let it harden. It's better for those bacteria that prefer less oxygen under agar. Spread plates involve putting an aliquot of diluted bacteria on a solid agar in a plate. Then you will spread the material uniformly with a sterile glass rod. The pros of these plate counts is that they count live cells. The cons is that they are cumbersome and often take a great deal of time. Serial dilutions. Why use serial dilutions? Simply put, because we do not know how many cells are in a sample. And why serial? Because serial dilutions are more precise and are an easier way to prepare. Here we have an example of a serial dilution, where subsequently smaller and smaller quantities or more and more dilute material is plated. Using serial dilutions, we're able to quantify how many organisms there are per mil in a solution. After incubation, we will count the colonies on the plates that have between 25 and 250 colonies or CFUs and use that to calculate the original cell number. The cell number is equal to the number of colony-forming units divided by the dilution times the dilution volume. See the example below for exactly how we calculate the number of organisms in the original solution. Next is the most probable number. In this direct method of quantifying organisms, some samples have too few CFUs for plate counts, or the environmental samples have water in them that would prevent a direct plate count. Sets of 5 lactose broth tubes are inoculated with 3 different volumes of a sample, as seen here, 10, 1, and 0.1 mils. The bacterial growth is then assessed through a change in color of the broth from red to yellow as lactose is fermented. The number of positive growths is compared to a table. This is an example of a most probable number experiment. In this example, the result is 5, 2, and 0, as seen at the bottom of this slide. Indirect methods. Turbidity is an easy way to evaluate or quantify bacteria microbial growth using spectrophotometry. The more cells, the more turbid or cloudy the suspension will be. This is very useful to monitor cell growth. In spectrophotometry, we can measure either transmittance or absorbance. Absorbance is sometimes called optical density. Transmittance is the amount of light that will actually pass through a specimen. The lower the percentage, the more cells are in the solution. Absorbance or optical density. The bigger the number, the more cells we have. The pros of spectrophotometry or turbidity studies is that they are quick and easy. The cons is it does not provide absolute numbers. It does not also differentiate between living and dead cells. This is an example of how we measure turbidity using a spectrophotometer. Metabolic activity and dry weight. More organisms equal more metabolic products. As an example, we might see acids or carbon dioxide produced in larger and larger quantities as the number of organisms increases. In direct proportion to the number of bacteria, these substances will go up. So, in metabolic activity studies, we simply measure products. Dry weight. The dry weight indirect quantification tool takes bacteria and literally dries them out. We remove the water and weigh them. This is often used in industrial applications. Other types of cell division can lead to an increase in cell numbers aside from binary fission. Fragmentation is done by cyanobacteria and actinomycetes, which give, in essence, live birth to cells through budding. Biofilms. Biofilms are microbial communities. They tend to form slime or hydrogels. Bacteria are attracted to biofilms by chemicals via quorum sensing. Organisms in biofilms will often share nutrients, and they can shelter one another from harmful factors. This is an example of the life cycle of a biofilm. Quorum sensing is a mechanism by which cells in a biofilm coordinate their activities in response to environmental stimuli. Chemicals called autoinducers provoke gene expression. Signaling molecules are different in gram-positive and gram-negative cells. Nutritional requirements are often a determining factor on how quickly or how slowly a population might grow. Early life on Earth had no oxygen available. Oxygen appeared as a byproduct of photosynthesis. As a result, there are organisms that prefer to live in oxygen-poor environments and grow best there, and vice versa. The oxygen revolution led to a massive extinction. There are damaging reactive oxygen species, or ROSs, that include things such as superoxide free radicals, hydroxyl radicals, and singlet oxygen. This is an example of organisms that grow under different oxygen conditions. The obligate aerobes, which are most resemblant of humans, can be found at tube A. The obligate anaerobes, B. These are organisms that cannot or will not tolerate oxygen. Facultative anaerobes, who prefer oxygen but can also live in environments without it. Aerotolerant anaerobes, who have really no preference, and microaerophiles, who are really only satisfied, happy, or content with very small amounts of oxygen. This slide summarizes each of the aforementioned test tubes. Some examples of organisms that fall into each category are listed here. Obligate aerobes include things such as Neisseria and Mycobacterium. Obligate anaerobes include organisms from the genus Clostridium. The culture of anaerobes is something that can be a bit of a challenge. The media and/or conditions must be free of oxygen. Oftentimes, a reducing media, including thioglycolate, must be introduced in order to drive oxygen out of the environment. Anaerobic jars and chambers will be employed to keep the environment oxygen-free. Detoxification of reactive oxygen species. Here we see some reactive and dangerous oxygen in the form of superoxide free radicals and peroxide anion. The superoxide free radicals must be converted to water by superoxide dismutase such that organisms are not destroyed by its presence. Peroxide anion must also be converted to water so that organisms can safely coexist with it. This is done by catalase or perioxidase in the reaction sequences seen below. pH can also affect the growth behavior of microbial colonies. Different organisms exhibit different preferences in terms of pH. Some organisms, known as acidophiles, prefer acidic environments, others alkaline environments, and others still neutral environments. Any violation in the preferred pH will affect the rate of growth of a species. Here we see some acidophiles. Lactic acid in bacteria can produce an acidic pH. Fermented milk products, pickles, and sauerkraut and pico de gallo are acidic foods that are preferred by acidophiles. Temperature can also have an impact on microbial growth. Psychrotrophs prefer cold temperatures. Mesophiles prefer moderate temperatures, and thermophiles and hyperthermophiles prefer hot temperatures. Like the previous two mentioned, osmotic pressure can also play a role in the growth of a microbial population. Osmotic pressure is found most commonly in hypertonic environments, or an environment where an increase in salt or sugar can lead to plasmolysis, where the plasma membrane will burst inside of the cell wall. There are, however, extreme or obligate halophiles that actually prefer this type of osmotic pressure for growth, and facultative halophiles which can tolerate it. Culture media. The culture media is the media upon which we prefer, or at least attempt, to grow microorganisms. Nutrients can be prepared for microbial growth in a variety of different culture media. Culture media are sterile if they have no living microbes. The organisms that we plate on culture media are referred to as the inoculum. The inoculum can be from a liquid, broth, or from a solid. Solid media contain agar, which is a complex polysaccharide. Solid media is used as a solidifying agent for culture media in petri dishes, slants, and deeps. The agar in solid media is generally not metabolized by microbes. That simply means they do not eat it. It also liquefies at 100 degrees Celsius and solidifies at about 40. Chemically defined and complex media. Chemically defined media is a media where we have knowledge of the exact chemical composition of the media. In complex media, extracts and digests of yeast, meat, or plants are used. These are a few examples of complex and defined media. Next, we have selective media. Selective media suppresses unwanted microbial growth. Here we have mannitol salt agar, which will inhibit certain types of bacteria that are not salt tolerant. Differential media is a media that makes it easy to distinguish colonies of different microbes. Colonies growing on differential media will often develop a unique appearance as a result of this growth. Here we see an example of blood agar that's used to differentiate streptococci. Selective and differential media is media that plays two parts or two roles for us. Here we have mannitol salt agar, which is selective for salt-tolerant microbes, and differential for mannitol fermentation. Staph aureus is a mannitol fermenter and will turn yellow. Simply put, this plate will suppress any growth of organisms that are not salt tolerant and then will change color to make identifying Staph aureus easier. And lastly, we have an enrichment culture. Enrichment cultures encourage the growth of desired microbes. As an example, we may assume a soil sample contains a few phenol-degrading bacteria and thousands of others. To isolate the phenol-degrading bacteria, phenol would be added to the medium, which encourages the growth of those bacteria and suppresses the others. And this concludes our coverage of microbial growth from Chapter 9 of your OpenSTAX book.